So welcome back to the lesson on replication of DNA. We were in the last class we have discussed a lot about the Mendelssohn-Stahl's experiment, and it is about trying to prove the mode at which replication occurs. We had three models. That is, the first one is dispersive, second is semi-conservative, and the third one is conservative one. Out of these three proposed models, it is shown by Mendelssohn-Stahl that the semi-conservative replication is correct. I'm not going into the technical details of the experiment, but the concept-wise, the parent double-stranded DNA molecule like this will denature and a new strand would be formed. Eventually, there will be formation of two identical double-stranded DNA molecules, and that is the semi-conservative replication. For this to occur, we have discussed about the substrates that are required. That is, they require DNTPs and a primer template junction. This is the template. The purpose of the template is to dictate the sequence of bases in the polymerizing strand. And the purpose of primer is to provide a 3 prime OH like this. Why is that required? Because DNA polymerase cannot initiate replication randomly anywhere. It can only do so when there is a primer template junction. Okay, that means DNA polymerase can only add more bases to this 3 prime OH. Quickly, if you are going to forget, this is deoxyribose 2 prime, this is 1 prime, this is the 3 prime OH. That is where polymerization happens. And the mechanism is that the bases will come that are complementary to the base that is present on the template, and the 3 prime OH on the primer will be activated, and the oxygen upon deprotonation will form an oxyanion, which will make a nucleophilic attack on the alpha carbon between alpha beta phosphate, and that is how it becomes a part of this phosphate backbone. The new incoming nucleotide becomes a phosphate, part of the new strand. So there are many things we have to check. Is the first thing is DNA polymerase is the enzyme that that polymerizes this reaction. So we did talk about parts of the DNA polymerase. We will also have some more in today's class too. So the DNA polymerase should be able to distinguish or discriminate between DNTPs from RNTPs. RNTPs are ribonucleic acid, nucleoside triphosphates that are subunits of RNA. Whereas in DNA, the nucleotides or the ribose is deoxyribose. Second thing is the complementarity should be accurate. That is, if this is the template, the correct complementary base should come and not any other bases. And then it then has to polymerize or make the incoming nucleotide a part of it by making the phosphoester bond. If there is a mistake, if there was a wrongly incorporated nucleotide, it should be removed. And then it should do it very fast. The genomes are usually huge. And then the replication should be very fast. Then we will also discuss about high processivity. In the last class, we did discuss about how DNA polymerase can distinguish between which ones, right now we are actually going to discuss about the correct base pairing. Okay, in the last class we did discuss some things, but let's get back to this. So if there is a correct base pair as shown in A, and if there is an incorrect base pair as shown in B, Say, for example, on the template that you have here, there is adenine, right? Adenine cannot base pair with another adenine. It can only base pair with T, like as shown in this case. If the base pairing is, if adenine base pairs can only base pair with T, and when that happens, the 3' OH there is a place within the DNA polymerase where the 3' OH can sit in like this and make some interactions. And here I hope you remember metal ion A and metal ion B. We discussed about that. And these will be in proper orientation, or spatially they would be arranged properly so that the groups can be activated, and then reactions can proceed. Say, for example, if there is a if the base pairing between them is not proper, if the base pairing between them is not proper, then the 3' OH is not within, is not close enough to make a phosphorus or nucleophilic attack on the alpha phosphate. So that kind of spatial constraints would occur if the base pairing is not proper. So the proofreading mechanism is very easy, or the checking whether the base pairing is correct or not is actually rather easy, because if the base pairing is not proper, further reactions would not occur because of spatial organization. Then we discussed about this. How does DNA polymerase distinguish between dNTPs and rNTPs? Or NTPs is fine. You don't have to say rNTP. The problem is they are very structurally similar, like last time we discussed. And second thing is And the second thing is that RNTPs are at least 10 times more concentrated than DNTPs. Then how does DNA polymerase fit together? Because the difference between RNTPs and DNTPs is very little. So the first difference is, of course, the 2' OH is here, as shown in B, and in A, it is a DNTP. In this case, in generally, there is a part of nucleic acid within the active site that are called as discriminator amino acids. If there is a OH, then the discriminator amino acids will make interaction with it, because of which further reactions would not have happened. Okay, or you can think of it that the discriminator region will not allow further activities to happen. And then the DNA will have an option, a DNA polymerase will get another chance. This nucleotide will be expelled and a new nucleotide will be taken up. And whichever base pairs correctly and whichever is not a ribose RNTP will proceed further into making this phosphodiester bond here. Okay, so I hope you understood two things, right? Whether how DNA polymerase chooses which one is there. You can think of the active site something like this. There are different types of RNTPs, there are different types of DNTPs. They keep coming and they keep going out. Whichever is not fitting properly into this, based on two things. What is it? First one is base pairing, and the second one is whether the 3' OH is present, 2' OH is present or not. If there is no 2' OH, then it is deoxy DNTP. And if the correct base pairing is there, that will get incorporated into the DNA molecule. Otherwise, it will be expelled. Something like that will happen. So you can understand at what speeds molecules will come in and go out, and the correct ones will be incorporated into the growing polymerase chain, growing nucleotide strand there. So I hope you remember that we discussed also yesterday that DNA polymerase can be, you can put your right hand in this shape, and this is the thumb region, palm region, and then the fingers region. They're just for your for our understanding. So the DNA Primer template junction is usually, primer template is like this. And this is actually the junction where you would have the 3' OH, and the rest of the template is not into the palm region. Only the one base to which a new nucleotide or the complementary base should be added is exposed in the active sites that's present in the palm region. We also discussed about metal ion A and metal ion B. Those two are important for the generation of oxyanion. Metal ion A will deprotonate, because of which it becomes a oxyanion and make a nucleophilic attack on the alpha phosphate. So the responsibility of metal ion B, along with other residues, aspartic acids, aspartates, is that they will keep these alpha, beta, and gamma phosphates in proper orientation, so that the bond between O and phosphate, alpha phosphate, can happen. So there are many other things. So what happens in the last time discussion we were in the last slides, I was telling, if the base pairing is correct, okay, there are some amino acid residues. You can see something like tyrosine, lysine, and arginine. There are many other amino acid residues within DNA polymerase that also function in it. So remember that we are not trying to mug up all these things, but from a biochemistry point of view, or even in the evolutionary point of view, it's important to understand how structure can be dynamic, right? The enzyme DNA polymerase is in one way, but the internal components, based on the interactions with different types of bases and molecules, it keeps changing. And doing so, it is polymerizing the DNA in the most possible um, correct way, right? So this is the O helix of DNA polymerase, which is normally like this. If the OH, if the base has come here, it was able to make proper hydrogen bonding, then this will form a closed complex, or it kinds of, so tyrosine has a phenyl ring, and it's a, I'm not drawing all, but yeah, it is a OH containing, and it has a plane like this, as you're seeing here. And you also remember that the bases also have their own ring structures, right? I'm just putting it, so in that case, it'll be roughly. So there are planes. I hope you remember what is base stacking. Base stacking is the interaction between these planes of these rings that are present as a ladders. The same way, this tyrosine, the ring structure of the tyrosine, can also have an interaction with the incoming nucleotide. And then the lysine and arginine residues, which are basic amino acids, can interact with the beta and gamma phosphates here. And so there are multiple mechanisms of how a base that is correctly, if it is a wrong base, it will be expelled. If it is not base pairing properly, it will be expelled. If it is a ribonucleotide RNTP, it will be expelled. But if a correct one comes, then there are mechanisms of how reactions will proceed forward. We have discussed about the metal ions, metal ion A and metal ion B, and then now we see some other, the O helix that will stabilize the interaction, and then the phosphodiester bond is made. Okay. So the path of the template, that is you can think of it as this template is rotating like this. As it is rotating, the template will be moving in this direction. I hope you have seen the videos that I have already uploaded please go to the replication videos and watch. Whatever mechanisms I'm teaching is already there, and it will better your understanding. So this is the incoming nucleotide, and the only base that is actually exposed in the active site is the base after the first base that is still in the single strand region. Okay, now the base will come and the new base will be polymerized to this DNA strand like this, and then you can expect a little bit of turn of this DNA like a spiral, and so the next base will come into the action to the active site. That is how DNA polymerase will keep polymerizing. So there are actually different types of polymerases. There are different types of polymerases, which are actually they have different functions within E. coli. Okay, I'm sorry about this. Yes, and I would want to discuss a bit on this one because okay, let me talk about this first. So E. coli has different types of DNA polymerases. You call them DNA polymerase 1, Pol 2, Pol 3, and there are also Pol 4 and Pol 5. Okay, we will discuss a bit of these in many occasions, especially when it comes to DNA repair and also as a part of PCR. So as we will the genes and all you don't have to mug up, but what I want you to remember is the rate of polymerization. DNA polymerase 1 can add up to 20 bases per second, and polymerase 2 can add up to 40. And look at Pol 3, it can add up to 1000 bases per second. That means you can understand the rate at which it is actually working. And the polymerization is always from, I hope you will remember that always, polymerization of even DNA or RNA is always from 5 prime to 3 prime. That means the chain grows from, because I told you there should be a 3 prime OH. Only then a DNA can add something else here, right? So the growth of the polymerization always happens from 5 prime to 3 prime. So because of which the opposite strand or the complementary strand would have been something like this. Okay. So there are some mechanisms that we need to discuss. I think it will come in a few slides. 3 prime to 5 prime exonuclease activity and 5 prime to 3 prime exonuclease activity. We will discuss that as we go. But now we will first talk about what is processivity. Processivity is something like this. You have a substrate as shown here, and you have a polymerizing enzyme here. And the, the, the point of it is how many bases does an enzyme add before it dissociates from the primer template junction. So if an enzyme is able to add only 3 and it dissociates, then you call it as non-processive, or its processivity is very less. Processivity is the ability to the number of subunits it can add before it dissociates from the substrate. Whereas a DNA polymerase that is highly processive can add thousands of bases before it dissociates from the primer template junction. Now I will want you to compare the processivity of these 3 DNA polymerases. DNA polymerase 1 only 200, polymerase 2 1500. That means it will add 1500, at least 1500 bases before it separates from the primer template junction. Look at DNA polymerase 3. It can add about 500,000. bases before it separates from the primer template junction. Okay, so DNA polymerase 4 and 5 are actually not so great. But they are still important. We will discuss that later on. I also want you to know what you can figure out about exonuclease. What is exo mean? Exo is outside. Endo would have been inside. That would be endo. This would be exo. Nuclease, that would mean ability to break. Okay, if it is breaking from the outside like this, then you would call as exonuclease. If it were breaking in the middle of the strand or DNA molecule like this, you would call it as endonuclease. Okay, so there are the different DNA polymerases, Pol1, Pol2, and Pol3. They have the exonuclease activity, which is important for proofreading. We will get to that later, but just remember those. I will skip this. We will go back. This is where I want you to discuss exonuclease activity. Say, for example, if you didn't understand in this slide, it's all right. Relax. We will have other ones to find out. So this is the DNA molecule. This would be the primer, and this is the template. And if I were, say, for example, I'm going to write some sequence here, and I'm going to now add a different sequence on the template, on the primer. So T A G C T T G C. So DNA polymerase has added the bases here. There is something troublesome here, isn't it? There is a mistake here. This is not base pairing. Although DNA polymerase has the ability to check if the base pairing is correct or not, but because, say, something like DNA polymerase that is adding about 500,000 bases, or say 1,000 nucleotides per second, there will be some mistakes. And in that case, what can the DNA polymerase do? So DNA polymerase are self-correcting as well. That means they can correct their mistakes, unlike many of us. So the DNA polymerase that was adding, it can interact with this C. It will notice that there is something wrong about it, right? It's based on everything, although I say it as in an anthropomorphic tone, it actually just means the interactions. We'll get to that. So it will cleave this base. It will cleave it and expel it from the medium, from the template. So in what direction is it cutting? This is the 3' end, and this is the 5' end of the growing strand. So what kind of exonuclease activity? Is it exonuclease? Yes, because it is breaking this bond here and expelling this C. So what is it? Is an exonuclease. First thing is it's exonuclease, and is it 5' to 3' exonuclease or 3' to 5' exonuclease? I think most of you could guess it, that it is 3' to 5' exonuclease activity. Okay, so this is the activity that it has. Now let's see it, try to understand the structural basis of DNA polymerase, polymerization activity and exonuclease activity. So here is the thumb region, here are the fingers, and here is the palm region that you have, and that's the active site. The exonuclease activity is present somewhere in the bottom of the palm, you can think of it, or the hand. If there is a mispaired base, that is a wrong base has been incorporated, like I have drawn here, then there is going to be the hydrogen bonds are not going to be proper, because of which the this whole strand, this whole DNA molecule will not be able to make interactions with the palm region of the DNA. So that means what is happening? The DNA can move forward and backwards, or it's not so tightly bound to the palm region. If mismatches are there, that is what happens. Then the DNA molecule can move into this position or back. And every time, this is where the nuclease activity active site is, right? So when it comes here, then using the exonuclease activity, the misincorporated bases will be eliminated. And then, because there are no misbase pairings, the interactions with this DNA polymerase, with DNA with that of the palm region will increase, and then polymerization will resume. So it is all about the amount of time. Say, for example, this is the polymerization active site, and here is the exoactive site. DNA can be in this place and started rapidly, it can keep growing, and if there is a mistake, it will move here because the interactions with the palm region have reduced. And in the exonuclease activity, the misbase paired base will be removed from the growing strand. And when there are no misbase pairs, the interactions of the DNA will increase with the palm region, and it will get back to the polymerization active site, and polymerization will happen fast. Okay, that's a fantastic mechanism of self corrections. So this is representing the same thing. I hope some people would like this picture. That you can think of it as one of it is the pol active site, and here as the exonuclease active site. It is clear it is 3' to 5' exonuclease active site. So when a wrong base has been incorporated, it will move into the exonuclease site. That is where it's actually removed. So please understand that the image shown here is much more schematic compared to what is shown in the previous one. This is from a different textbook, Biochemistry Leininger, I guess. So this illustration is for better understanding only, okay? It's not very representative, unlike the previous one that is shown. So if it has incorporated, it will move to the exonuclease activity, and there the wrong base, mispaired base will be removed, and then again DNA will start from will be in the polymerization site, and then polymerase will happen. So there are now we are actually getting into the mechanistic ways of how the DNA polymerase actually works, or the whole replication happens. So we learned about the double-stranded DNA. This is the or the semi-conservative replication. It's the same picture. You can think of it. This is the double-stranded one. This is where this region is where denaturation has occurred, and this is the DNA where replication has occurred. And now we are able to see one DNA giving rise to two DNA strands. The structure here is described as the replication fork. So you can assume that in CIA or in any semester, it's a very likely question that you draw the structure of replication fork. Not necessarily in the coils. You can put it straight lines, but you should be able to draw it and label them properly with the ends, all the 5' ends and 3' ends properly indicated as shown here. So polymerization, the direction of polymerization is always 5' to 3'. I have said this before. Okay. And we have two strands. I will try to probably use I will use an empty slide so that I can draw it the way I want you to represent in the future. So I'm trying to use this as a way so that you will be able to draw the picture in a similar way. I'm only putting one side, so I'm not drawing the helical structures and so on. This is double-stranded part, this is the denatured part, and this is where application is happening. I will put the colors differently for the new strands. This is the new strand that is being synthesized, but let me first put, if this is 5' and this is 3', this part would be, this is 5', and for this strand, this will be 3'. Replication can always happen from 5' to 3'. Not replication, you should say polymerization. Okay, keep that in mind. So we also need, for polymerization to occur, DNTPs, assume they are there. We also require a primer because DNA polymerase requires a primer template junction for initiating replication. So first thing, I will take this strand, which is easier. So I'll draw a primer. Primer is usually indicated like this. And if there is a primer and there is OH, what will DNA polymerase do? DNA polymerase will, I'm trying to find a different color. So DNA polymerase will come and add the, will start polymerizing. Is that right? That is wrong. Why is that? The complementary strand, the primer, would have been 5' to 3' like that. Yes, I hope you understood that. Polymerization can only happen in this direction, not this direction. So I'll erase it and I'll start with the easier one that should be the opposite side. Sorry. Okay, this is easy. Here is 5' and this one is 3'. Now I think I'm in line. So there is always scope for confusion, so keep track of it. So DNA polymerase will start adding bases and it will keep on adding. And as it is adding, more and more DNA will get denatured. More denaturation will occur, right? These are the hydrogen bonds that are present. Then these strands will separate like this, denature. And then DNA polymerase will keep on adding more and more. And this is referred to as the leading strand. There is another, the problem is discussed looking at the lower strand. The problem is you have a primer here, say, for example, and now polymerization should occur. How would that polymerization happen? There is a problem because we have 5' here, 3' here. Polymerization always happens 5' to 3'. So DNA polymerase can only add in this direction, right? And more DNA will open up, and assume that I'm going to draw a new primer here. A new primer will come and attach there, and DNA polymerase will take this up and will produce only till here. And again, as more and more DNA gets denatured, a new primer is synthesized and a new fragment is produced like this. Each of these fragments are called as Okazaki fragments. And it's also this strand where it is called as the lagging strand, because it is happening discontinuously. Whereas look at the leading strand, it is happening continuously. It will keep on adding like this. It is continuous. Okay. So when we are drawing things, when we want to have a replication fork, I hope if it were on board, we could do it better. But even on paper and pen, I hope you will be able to do it better. So you should be able to draw straight lines like these and be able to indicate the n groups polarity or polarity of the DNA strands that are being made and the location of the primers and the newly synthesized strand. Say if you're doing it, I hope you look at this point. This part is DNA, and I'll come to the primers in a while, but you should not join it like this. That is wrong. If you have joined it, that means it is continuous DNA molecule, which is actually not inaccurate. Okay, so don't connect it continuously like that. Okay, what I'll do now is we will just have a quick review, and then I'll give you introduction for the next class, and then we'll stop. So I hope now you can see what I have already described in the previous slide. I'm just going to use the same uh, because this is from the textbook. It will be nicer. Direction of replication is in this way, right? That means more and more DNA is getting separated or denatured, and then more and more is synthesized. The polymerization is always 5' to 3'. That is, you can see in the leading strand, the top one. Okay, so this is the leading strand that is being generated, which is continuously polymerized. Whereas the bottom strand is the lagging strand. It is produced in discontinuous maze, and the small fragments that are formed they're not very small. They are sometimes there are hundreds of bases, but still they're relative to the genome, they are small. They are called as Okazaki fragments after the discoverer of them, Okazaki. And lagging strand produces discontinuous DNA molecules referred to as Okazaki fragments. The problem here, if you look at it as mechanistically, DNA, this is the direction of replication. Replication fork is moving in this direction. So for lagging strand, polymerization is in this direction because it is synthesizing from 5' to 3'. No issues at all. Look at the lagging strand. The direction of replication is this way, and the direction of polymerization is in this way. That's kind of contradiction, isn't it? Some of the things the nature solves is amazing. I will use the next slide to indicate that, to discuss just that part. So what I want you to know is here we have shown DNA polymerase as two independent subunits, but that's not how it happens. They are clamped. It's a much bigger complex enzyme, multi-enzyme complex. So they are actually joined. And when they are joined, how can the replication be, how can the overall direction of the replication fork is in this way, and the direction of polymerization is in this way? How is it possible? That is something that DNA replication has solved, and it's beautiful in that sense. We will discuss that. Okay, it's not. The first thing is the primers. Where do primers come from? DNA polymerase can only add bases to 3' OH. And these primers are produced by an enzyme called as primase. Okay, primase produces primers. And these primers are usually, they are made up of RNA. So if you take one of the difference between DNA polymerase and RNA polymerase, DNA polymerase and RNA polymerase. RNA polymerase is can start polymerization without the need of a primer. It does not require a primer, whereas DNA polymerase requires a primer. So the first thing is primase is an RNA polymerase. It is an RNA polymerase which is usually associated with replication only. That's why we give a special name called as primase. It synthesizes a small RNA molecule like this in using the template here. Its processivity is not big, not high, so it releases. Only it produces a small RNA molecule like that and leaves. Then DNA polymerase takes it up and synthesizes the rest of the DNA there. So the end products of this, for example, is that in the lagging strand, you have several regions of RNA molecule. It's not DNA. This is also RNA molecule. Okay. So we have an enzyme called as RNase H. H stands for hybrid. What is hybrid here? This part of it is DNA, and in this part is RNA. So that's the hybrid. RNase. What does the enzyme name say? It degrades RNA. And which RNA? RNA that is present in the hybrid. So if it has RNA like that, it cannot do anything. If there is RNA as a double strand, it cannot work much. But if there is a DNA and RNA, that's a hybrid, then it comes and cleaves away the RNA molecule like from here. There are different types of RNases. Some RNases will act on any of these. Some will act on only single-stranded RNAs. Some of them will act on double-stranded RNAs and so on. This particular RNase H will works on the primers that were synthesized during replication. It will degrade this. And then there is a gap that is produced, and that gap will be used by DNA polymerase because this is a primer, and you have a primer and you have a template. DNA polymerase fills up the gap. And every time the last base is there, that is called a nick. I hope you remember what is a nick. I described it to you when we were discussing about it. When all the base pairing, everything is right, except there is one phosphate bond that is not made, that is called a nick. So DNA ligase is an enzyme that seals or makes this phosphate backbone continuous. It seals that small gap. So how does DNA that is present unwind or denature? For that, there is an enzyme called as helicase. The helicase is the enzyme that will break the hydrogen bonds that are present here, and then these two DNA strands are separated. If the two DNA strands are separated, but they are complementary, they should be able to form base pairs again, right? To prevent that base pairing, renaturation of those, we have, and these are called as SSBs. Full form is single strand binding proteins, right? In the previous slide, helicase is fine. It is just unwinding and breaking the hydrogen bonds. It's doing a lot of work. And after going for a distance, there is always a spontaneous way of these can hydrogen bond and renature. That is prevented by single strand binding proteins. And then we discussed about topoisomerase. It has to do with supercoiling. I will take that up in the next class. But what I want you to understand is I will use this space for it. So because I think it will, when I discuss this particular concept twice, it will be better. We have DNA polymerase, and This is the replication fork. We said, sorry, this is 3 prime, right? And the DNA molecule being synthesized is this 5 prime, 3 prime. That is the leading strand. In this bottom one, we have problem that is going in this direction. So polymerization can only happen here, whereas the movement of the replication fork is in this direction. I am talking only in terms of lagging strand. Leading strand does not have any issues. It is continuously produced, no problems, because the direction of polymerization and the direction of replication fork going further is in the same direction. So there is one way of how nature has solved this kind of puzzle, and that is done by um, a simple or ingenious in a way. I can think of it that way. Now I will draw the same. I'm reducing the size to fit it in. And now I'm going to do something about the template of this template of the lagging strand. I'm going to put it in this way. I bent it. Okay, I made a loop. So 3 prime, this is 5, this is 3, and this is 5. I just bent it. I hope you will adjust with the drawing. Now, if I had a primer here, okay, which side should it polymerize to? The primer would be, this will be the 5 prime. If it is not clear, don't worry. We will discuss in the next class as well. You can consider this as the primer for it. 3 prime and 5 prime and 3 prime. Did I put it properly? If you have any questions, you can tell me now. Because this is the template that we have, and to the template I have drawn this primer. And primer will be in opposite orientation. That is 5 prime to 3 prime. Now, if polymerization happened, it should happen in this direction. Right? What is the direction of replication overall? This way. What is the direction of polymerization now? It is also in this way. And that is how two of these DNA polymerases working together, they are linked as a multi-enzyme complex, and they can still make this polymerization happen. Okay, just stay online. Trying to find something, or you can try watching this. So this is, yeah, 1 second, sorry. This is the most amazing animation I had for a while. I hope you are, you cannot view it, I guess. You cannot listen to the audio. I will post the links. You can always watch them. And this is DNA molecule that is present here, shown. This is histone molecules. We will read about all of these. And here is gene transcription happening. RNA polymerase is reading the DNA and producing new RNA molecules like that. This is the nuclear pore complex. Once the RNA is produced, it is sent out through the nuclear pore complex. All replication and transcription happens within the nucleus in eukaryotes. In prokaryotes, no issues of that. This is the DNA double-stranded. It is shown as that. So if you take one DNA from our one cell, the total, it will be around 2 meters. It's longer than most of us, I guess. But because it has to fit in a very small structure, about 10 microns or so, it is wound around nucleosomes, and a lot of supercoiling is involved. That is why I did even touch about DNA topology. And then they will form into complex structures. We have a whole lesson on that. We will discuss about it. And then they will form something like structures, chromosomes that can be observed during metaphase. I hope you're also good enough or you have basic idea about cell division. I'm trying to look for the DNA polymerase. Yeah, okay, it will come. So during metaphase, that is how the chromosome structure. This is a real microscopic image of chromosomes segregating into during anaphase into two um, daughter cells. And that is anaphase. Now you see that there's two poles that have separated DNA like that. And now cytokinesis, and we have two different cells. And here is, I want you to understand this. This is the structure I was telling about. All this comes to this. This is DNA replication that is happening. And this is the DNA that is being replicated. This is the whole DNA complex, the multi-protein complex that includes DNA polymerases also. You see these blue ones that are shown, they are the DNA polymerases. And I hope you can guess, this is the DNA polymerase here and here. There are the two DNA polymerases. And they keep continuously making these loops so that, and why do they make those loops? To keep the lagging strand, the polymerization on lagging strand in the same direction of the direction as that of the replication of the DNA. We are seeing this at a very, very low rate. This is an animation. This is not the real image. People, some scientists got together and they tried to use real structures and made an animation out of it. And that is how it would look like. Okay. I hope you understood the structure and the complexity, or actually the beauty of how well this can be orchestrated. Just watch it once more, and then we will continue with the next class. Please expose yourself to more and amazing stuff that happens. There are two things that are amazing and beyond our perceptions. One of it is too far in the space that we cannot see, right? Or we cannot probably understand. And the second one is too small in space, too small objects that we cannot see. And at least one way is all this DNA polymerase, replication, transcription, all of biology that we are actually studying is happening in our body right now. And we need sophisticated instruments to see, and that's not always a possibility. And then the only way is to be able to watch these kinds of animations to broaden our perceptions or imagination about how great these things can happen. And these are happening just by themselves, right? Provided the conditions, this is kind of an automatic thing that happens. So use the YouTube as well as possible to ensure your views, your ideas, or imagination is expanded. The rest of the things will come to you automatically. Okay, use the technology for increasing your own science and knowledge. Any questions? I think. Anybody has any questions?
So just one question.
Yes.
When we make this loop, there would still be fragments, right? The thing would still remain fragmented. We cannot avoid the fragments, right?
That's right. That's right.
So the only purpose is to make sure that the replication and the action of the enzyme is in the same direction, right?
That's right.
That's all.
Yes.
Okay.
Otherwise, you would need two independent DNA polymerases to carry out that side one and this side one.
That is if it is not looped, right?
Yes, yes. If you
Okay.
don't
Thank you.
want, if you want it continuous, that will be the problem.
All
But
right.
so
Thank you.
come up with, I'm okay with the fragmented molecules. I can repair, remove all those stuff, right? To come up with a new one. So new one will become haphazard. One thing about replication is all of replication should be very coherent. If you make mistakes, you're producing too much DNA. And that DNA might, the stability of the DNA itself will go off. I will want to discuss that at a later time. But remind me if I didn't do that. I will discuss about it in the next class. One
All right,
thing
sir.
is DNA replication should be highly orchestrated. It should not happen whenever possible, wherever possible. It should happen only at the accurate time. That is one of the reasons why DNA polymerase is dependent on a 3' OH. It requires the primer, right? If it is not, it would initiate replication anywhere possible, and that would complicate the stability of DNA itself. Okay? But
Yes, sir.
we'll have a discussion as we progress through. Yeah.
All right, sir.
Thanks for the
Thank you.
question. Yes. Anybody else? Okay. Thank you. I'll see you in the next class. Bye bye. Have fun.
Thank you, sir.
Thank you, sir.